have you ever wondered, like how scientists actually figure out why some diseases seem to run in families? I mean, we're talking billions of pieces in the human genome. It's like the ultimate needle in a haystack problem. Welcome to the deep dive. Today, we're really getting into something fascinating. Genome-wide association studies, or GWS, as they're usually called. And this isn't just some dry acronym. It's a revolutionary approach. It completely changed how we understand complex diseases, things like type 2 diabetes, heart disease, you name it. It's all about finding these tiny genetic signposts hidden in our DNA, the ones that kind of whisper clues about our health risks. So our mission for you today is to give you a really clear picture of how these huge studies work, how scientists cleverly overcame some, frankly, massive early challenges, and where this whole field is heading next. It's a real journey, like detective work, constantly improving the tools.
Yeah, what's so fascinating about GWS, I think, is it's how it keeps evolving. It's this amazing mix of technology, really clever statistics, and deep biology, all pushing each other forward. It's been quite a ride.
Okay, so let's start at the beginning then. Before GWS came along, geneticists were kind of like family historians, right? Tracing diseases down through generations.
Exactly. That approach, linkage analysis, was brilliant for what we call Mendelian diseases.
Ah, those are the ones caused by a single gene with really clear inheritance, like cystic fibrosis.
Precisely. Huntington's disease is another classic example. Linkage worked really well there, but it hit a wall with the common complex diseases.
Why was that?
Well, things like diabetes or heart disease, they just didn't follow those neat single-gene patterns. The genetic picture was clearly way more complicated, needed something else entirely.
Right. And that led to this big aha moment, didn't it? The idea that common diseases probably weren't down to one big genetic fault, but maybe lots of common genetic variations.
Each having just a tiny little effect. Yes, that was a huge shift in thinking about the architecture of disease.
Okay, so if that's the hypothesis, you suddenly need a way to actually test it, right, across the whole genome.
Exactly. The old methods couldn't handle that scale or subtlety. You needed a totally new methodology.
And almost like magic, the technology started catching up just when it was needed. High throughput genotyping, specifically those single nucleotide polymorphism arrays, SNP arrays.
Yes, SNP arrays were absolutely crucial. These chips let researchers quickly scan hundreds of thousands, eventually millions, of these common single-letter DNA variations across many people. It made the genome-wide part feasible.
But you need more than just the tech, you need a map, right?
You do. And that's where the HapMap consortium came in. It was this massive international effort to catalog common genetic variants, how frequent they were, how they were correlated in different populations. It provided the essential reference map. Without HapMap, the SNP data would have been much harder to interpret.
So you've got the theory, the tech, the map. And then 2005 happens. The first big landmark GWS papers start coming out.
That's the milestone year, yeah. Screening whole genomes, large groups of people, cases and controls, looking for those statistical links between SNPs and disease.
But it wasn't just a straight line from A to B, was it?
Not at all. It was much more like a dynamic feedback loop, as some have called it. The biology ideas pushed the tech and stats forward, and then the results from those studies refined the biological ideas. It created this real acceleration in discovery.
Okay, let's get into the nitty-gritty then. How do these studies actually work? You said it's a mass univariate approach.
Yeah, basically, in the simplest form, you test each genetic variant, each SNP, one by one, independently. You're just asking, is this specific variant statistically associated with the disease or trait we're interested in?
Like a correlation hunt repeated millions of times.
Pretty much. Early on, simple tests like Pearson's chi-squared were used, comparing genotype counts in cases versus controls. Does this group have more of a certain variant than the other? But chi-squared doesn't really look for a trend. Like, does having 2 copies of a risk variant increase risk more than having 1 copy? So the Cochrane-Armitage trend test became popular because it specifically looks for that kind of dose-response relationship, more powerful for many genetic effects.
And then came the real workhorses, which are still central today, linear and logistic regression.
Absolutely. Regression models offer way more flexibility. Linear regression is great for traits measured on a continuous scale. Think height, blood pressure, cholesterol levels. It estimates the effect size, the beta value, for each additional copy of the risk allele.
Right.
And for binary outcomes like do you have the disease or not. Logistic regression is the tool. It models the probability of having the disease, usually expressed as an odds ratio.
Yeah.
It's statistically more appropriate for those yes-no questions.
And the beauty of regression is you can add other things into the mix.
Exactly. You can include covariates, age, sex, environmental factors, whatever might confound the results to adjust for them. Plus, you can test different genetic models, like dominant or recessive effects, not just the additive one. Super versatile.
So you run these millions of tests.
Yeah.
You get millions of results, like p-values. How on earth do you visualize that? How do you make sense of it?
Yeah, that's where a couple of really key plots come in. They became iconic, really.
The skyline plot.
The Manhattan plot, yeah. Named because it looks like the Manhattan skyline. The x-axis is the position along the chromosomes,
Yeah.
genome-wide. The y-axis is the statistical significance, usually the negative log base 10, of the p-value. So higher points mean stronger associations.
And those really tall skyscrapers are the SMPs that cross the significance threshold.
Precisely. They jump out at you.
Mm-hmm.
The other crucial plot is the quantile plot, or QQ plot.
What does that one show?
It compares the distribution of your observed p-values against the distribution you'd expect just by chance.
Yeah.
Ideally, most points should fall along a straight diagonal line.
And if they don't, they curve upwards.
Ah, that's the warning sign. An upward deviation, especially for the less significant SMPs, indicates inflation. It suggests your test statistics are systematically higher than they should be. It was an invaluable early signal that something else was going on, like confounding.
Okay, so those QQ plots were basically flashing red lights saying, hold on, maybe not all these signals are real, which leads us right into the challenges. So the first GWAS results were pouring out, super exciting, but then people started realizing, wait a minute, some of these associations might be false positives. What was messing things up?
Well, a really big one was population stratification. This happens when you have subgroups within your study sample that have different genetic ancestries.
Okay.
And if those subgroups also happen to have different rates of the disease you're studying just due to environment or other factors, you can get a spurious association. It looks like a SMP is linked to the disease, but really it's just marking ancestry, which happens to correlate with disease risk in your specific sample.
Ah, right, a hidden confounder. So how did they fix that?
Early on, there was a simpler method called genomic control, or GC. It basically calculated an inflation factor, lambda, from the QQ plot and used it to adjust all the test statistics downwards. Kind of blunt, but helpful.
But then came something more sophisticated.
Yes, principal component analysis, or PCA. This became, and still is, the standard. PCA looks at genome-wide SNP data to find the major axes of genetic variation in your sample, which usually correspond to ancestry.
Like finding the underlying genetic map of your participants.
Kind of, yeah. You calculate these principal components, PC1, PC2, etc., and then you include them as covariates directly in your regression model. Programs like Eigenstrat pioneered this. It very effectively controls for those ancestry differences.
Okay, so that tackles differences between groups. But what about hidden similarities within the group? Like people being related without you realizing?
That's the next puzzle, cryptic relatedness. Standard GEOS methods assume everyone in the study is unrelated. If you unknowingly include, say, cousins or even siblings,
It violates the math.
it violates the independence assumption exactly. Related individuals share more DNA than expected by chance, which inflates the test statistics, potentially creating more false positives.
So how do you account for Uncle Bob showing up unexpectedly in your data set?
You have to explicitly model the relatedness. You calculate a kinship matrix, sometimes called a genetic relationship matrix, or GRM. This matrix estimates the degree of genetic similarity specifically, identity by descent, or IBD, between every pair of individuals in your study.
So you map out the whole hidden family tree, genetically speaking?
In essence, yes. And then you incorporate that matrix into your statistical model to properly account for that non-independence.
Okay, that makes sense. Now on to another big head scratcher from that era, missing heritability. What was that all about? It sounds like genetics just wasn't working as expected.
It was a really interesting period. We knew from twin and family studies that many complex traits had a substantial genetic component, high heritability. But when the first GWAS results came in, the handful of SNPs they found only explained a tiny fraction of that expected heritability.
So where was the rest of it? Was the genetic effect just missing?
That was the question. But the crucial insight looking back is that it wasn't really a biological mystery, but more of a statistical or methodological one.
How so?
Early GWAS, with its stringent significance thresholds, could only reliably detect common variants with relatively large effects. But the thinking shifted towards that many small effects model we talked about earlier. The heritability wasn't missing. It was just spread out across thousands, maybe tens of thousands, of common variants, each with a tiny effect below the detection threshold of those early studies.
Ah, so the signal was there, just too diffuse to pick up easily.
Exactly. And the resolution didn't come from finding some new type of gene, but from a change in statistical approach. Methods like genome-based restricted maximum likelihood, or GREOM, came along.
What did Dreamall do differently?
Instead of asking which individual SNPs are significant, Dremel asks, how much of the total variation in the trait can be explained by all the SNPs together, even the ones that don't reach individual significance?
So it looked at the collective effect.
Right. And suddenly, for traits like height, the amount of heritability explained by common SNPs jumped from maybe 5% to closer to 50% or more. It showed the heritability was there all along, captured by the SNP arrays, just distributed differently than initially assumed.
Wow. So dealing with stratification, relatedness, and understanding this missing heritability, it feels like all these solutions were converging.
They absolutely were.
Hmm.
And they converged on a single, powerful statistical framework, linear mixed models, or LMMs.
Okay, LMMs, what makes them so powerful?
LMMs are kind of the unified theory for these problems. They can simultaneously account for both population structure and cryptic relatedness within one model.
How do they do that?
They do it by including what's called a random effect in the model. This random effect uses that genetic relationship matrix, the GRM we talked about, to model how the similarity in phenotypes between people relates to their overall genetic similarity. It explicitly accounts for that shared genetic architecture.
So it builds the relatedness and structure right into the association test itself.
Precisely. It elegantly handles both major confounders at once. And software like GCTA and later Bolt LMM and Fast LMM made running these complex models feasible even on massive data sets, hundreds of thousands, even millions of people.
That really feels like a turning point, moving away from just looking at one SNP at a time to a much more integrated whole genome view.
Definitely a major leap forward in robustness and accuracy.
Okay, so with LMMs handling the big confounding issues, the field could focus on refining the picture. Now, running millions of tests, you're bound to get some flukes, right? How do you separate the real signals from just random noise?
Yeah, that's the classic problem of multiple hypothesis testing. If you set your significance level at the standard p 0.05 and run a million tests, you'd expect 50,000 significant results just by chance.
Whoa! Okay, clearly need to adjust for that.
Right, the simplest way is the Bonferroni correction. You just divide your desired alpha level, say 0.05, by the number of tests you ran. So for a million SNPs, your threshold becomes incredibly tiny, like 5 by 10 to 8. That's the standard GWA significance threshold now.
Super strict.
Very strict. It controls the family-wise error rate, FWER, the chance of getting even one false positive across all your tests. But it's often seen as overly conservative
Why's that?
because it assumes all your tests are independent. But SNPs near each other on a chromosome are often correlated due to linkage disequilibrium, or LD. They aren't independent tests.
So Bonferroni might be throwing out some real signals.
It might be, yeah. So another popular approach is to control the false discovery rate, or FDR. Instead of controlling the chance of any false positives, FDR controls the expected proportion of false positives among all the results you declare significant.
Ah, so it allows for some false positives but keeps the overall rate low.
Exactly. It's generally less stringent than Bonferroni and often more powerful, finding more true associations while still controlling errors in a meaningful way. It's about finding a practical balance.
Okay, so you found a statistically significant region, but GD often points to a whole block of SNPs that are correlated, right? Not necessarily the single causal one. How do you zoom in?
That's the job of fine mapping, specifically Bayesian fine mapping methods nowadays. The goal is to dissect that associated region, that LD block, and figure out which specific variant, or maybe small set of variants, is most likely the one actually driving the association signal.
How does it do that? Does it use biology?
It mainly uses statistics, actually. It considers the association strength of each SNP in the region, the LD structure, how correlated they are, and sometimes prior biological information, to calculate a posterior probability of causality for each variant.
And the output isn't just one SNP.
Often the output is a credible set. That's a small group of highly correlated SNPs that together have a high probability, say 95% of containing the true causal variant. It narrows down the possibilities significantly for experimental follow-up. Tools like SUSY and FineMap are really good at this now.
Makes sense. And what about machine learning? We hear so much about AI and ML everywhere. How does that fit into GWatchers?
ML and deep learning are definitely being used, especially for tasks like prediction building models to predict disease risk, like calculating polygenic risk scores. PRS, which sum up the small effects of many SNPs.
But is it helping find the cause?
That's the key distinction. ML models are often optimized purely for predictive accuracy. They can be black boxes, they might predict well, but you don't always know why. In genetics, particularly for understanding disease, the biological interpretability is often just as, if not more important than pure prediction. We want to know the mechanism.
Right, you want to understand the biology to maybe find a treatment, not just know someone's risk score.
Exactly. So while ML is a powerful tool in the toolkit, especially for prediction and integrating complex data, there's still a strong emphasis on methods that provide biological insight.
Okay, so it feels like we've gone from just finding associations to really trying to pinpoint causality and understand function. That's a huge step.
It really is, moving beyond correlation towards causation.
So GDMAS has come a long way, tackling confounding, finding that missing heritability, refining the signals. But now it seems the field is realizing the genome itself is only part of the story. What's the next big step?
You're spot on. The genome is the blueprint, but disease often happens through changes in how that blueprint is read and used. So the next frontier is definitely about integrating genomics data with other large-scale biological data, what we call omics data.
Oh, it's like genomics.
Genomics is the start. But then there's transcriptomics, looking at RNA expression, proteomics, looking at proteins, metabolomics, looking at small molecule metabolites. The idea is to get a more holistic, multi-layered view.
Why is that so important now?
Well, a huge number of GWAS hits fall in regions of the genome that don't actually code for proteins. They're likely regulatory regions. So just knowing the SNP doesn't tell you what it does. Integrating omics can help bridge that gap, linking a genetic variant to changes in, say, gene expression, then protein levels, and ultimately to the trait or disease.
Connecting the dots from DNA to function. Let's break down some of those omics, transcriptomics.
That's about measuring RNA levels, which genes are being turned on or off, and how much. There are methods like TWS, transcriptome-wide association studies. They essentially try to predict gene expression levels from your genetic data and then associate that predicted expression with the disease. It provides a potential molecular link between the GW's hit and a specific gene's activity.
Okay, makes sense. What about proteomics? Proteins are the real workers in the cell, right?
They are indeed. Proteomics measures the abundance of proteins. It gives you a snapshot that's often closer to actual biological function than RNA, because RNA levels don't always perfectly correlate with protein levels.
Any examples where proteomics found something missed by other methods?
Yeah, actually, there was a neat example where integrating proteomic data helped identify an association between the gene CRE, BIB1, and bipolar disorder. Apparently, this link wasn't clearly seen just using transcriptomic data, highlighting that proteomics can offer unique and sometimes crucial insights.
Interesting. And metabolomics, that sounds like metabolism.
Exactly. It measures the levels of small molecules and metabolites, like sugars, lipids, amino acids. These are the end products of many cellular processes. Linking genetic variants to metabolite levels using tools like MetGWAS can be really powerful. Often, a genetic variant has a much stronger effect on a specific metabolite level than on the complex disease outcome itself. It can really help pinpoint affected pathways.
So it really is like moving from just looking at the blueprint to watching the whole factory operate, from the instructions to the machines to the final products.
That's a great analogy. It's a shift away from a purely reductionist view, just focusing on the SNPs, towards a much more systems-level, holistic understanding of how genetic variation actually leads to disease.
And the hope is that by tracing these paths,
The hope is that omics data provides that crucial missing link. You can trace the effect of a GWAS SNP through changes in RNA, then protein, then metabolites, potentially identifying the specific culprit molecules whose disruption leads to disease. And those molecules become much better candidates for potential drug targets. It turns the statistical hunt into a real biological detective story, aiming for therapeutic insights.
wow, what a journey we've covered today. It's amazing to think how far GWAS has come. We went from those early days, simple tests, linkage analysis.
Right, through tackling huge challenges like population stratification, figuring out the whole missing heritability puzzle.
To developing these really sophisticated, integrated models like LMMs, it's been quite the evolution in statistical genetics.
And as we discussed, the focus now is shifting so much more towards interpretation, function, causation, using things like fine mapping, and especially this exciting integration with multi-omics data. It's not just about finding dots on a Manhattan plot anymore. It's about connecting those dots into biological pathways.
So thinking about the listener, what's the big takeaway? Why does this deep dive into statistical methods matter?
Well, ultimately, this whole journey is about getting us closer to understanding complex diseases at a really fundamental level. And that understanding is the bedrock for developing better prevention strategies, better diagnostics, and eventually more personalized treatments. It's building that detailed map we need for precision medicine.
So the future really lies in putting all these pieces together, the genome, the transcriptome, the proteome.
Absolutely. The vision is for computational frameworks that can synthesize all these different data streams to really get that holistic systems-level view of how our genes interact with our environment to shape who we are and what diseases we might be susceptible to. It's about understanding the whole complex system.
It really makes you think. As we get better and better at mapping out this incredibly intricate interplay between our genes, our molecules, our health, how might that change our very definition of what it means to be healthy? What does normal even look like at that level of detail? Something to ponder.